from this, the chapter first is a validation. We have seen already the introductory part of chapter 1. Now we are going to see the chapter 1 in detail. Today's topic is analytical method validation. So, see, strategy for analytical method validation. The validation of a specific method must be demonstrated through laboratory experiments by routinely analyzing samples. So, see, we have seen a definition of validation already. Then, what is meant by method transfer? Then, the method transfer can be done from laboratory to laboratory. This is called as a interlaboratory practices. So, for that purpose, we have to validate initially the procedure, and then only we can transfer the procedure from one laboratory to the newly developing laboratory. So, see, suppose we have one procedure which is of a pH metry, which is developed by us only. So, this procedure rights are with us only, not with the others. So, we want to export this procedure to the another laboratory. We want to transfer this procedure of a pH metry to the another laboratory. For that purpose, we have to initially check the correctness, the truthness of that procedure. And this checking of truthness, checking of correctness, checking of exactness, this is called as a validation. So, initially the procedure we have to check its correctness, truthness initially. Then only we can transfer this procedure from our laboratory to the another laboratory, which is newly developed laboratory and which has know already the procedure. So, see, the strategy for analytical method validation is as the validation of a specific method must be demonstrated through the laboratory experiments by routinely analyzing samples. So, initially which procedure we have developed, we have to check the correctness of this experiment in our laboratory, and this correctness can be checked by routinely analyzing the sample experimentally. Then the preparation and the execution have to follow a validation protocol. So, we have a standard procedure. This is called as a validation protocol. The protocol is prepared and this can be executed, okay, preferably written in a step-by-step instruction format. So, see, you have performed so many experiments so that you have initially the procedure which is given to you and you have to perform this procedure. So, see what's written in the procedure. Aim of the experiments, chemical required for the experiments, then the procedure is there, then the result is there, plots are there, and the conclusion is there. So, this is a step-by-step procedure and this is called as a validation protocol. So, we have to prepare this protocol, execution of that protocol can be done, and then we have to perform the procedure step by step. Then this proposed procedure assumes that the instrument has been selected. So, obviously, procedure which is we have to measure the pH or we have to measure the EMF, the respective instrument will be available in our laboratory. So, if not there, then you have to purchase this instrument. So, suppose you have a procedure, then it is assumed that the instrument should be selected and the method has also been developed. Then it meets a criteria such as ease of use, ability to be automated and controlled by a computer systems, cost per analysis, sample throughput, turnaround time, environmental, health and safety requirements. So, see, we are going to develop a new laboratory. So, instrument should be there. Each and every requirement can be fulfilled initially, and then only we can transfer procedure from the already developed laboratory to the newly developing laboratory. So, each parameter should be there. Then, successful acceptance of the validation parameters and performance criteria by all involved parties requires a cooperative effort of several departments like quality control, analytical development department, regulatory affairs, and the individual requiring the analytical data. So, for the acceptance of the validation parameters and the performance criteria, there should be a cooperative efforts of several departments. As you know, any pharmaceutical industry can be with quality control department, research and development department, analytical department. Such types of departments can be cooperative. Then and then only there will be successful acceptance of the validation parameters and performance criteria can be done. Then the operating procedure or a validation master plan, this is called as a VMP. Operating procedure or a validation master plan must clearly define the rules and responsibilities of each involved department in the validation of analytical methods. So, operating procedure or a VMP clearly defines the rules and responsibilities of each department which is involved in the validation procedure. Then the actual part is here, the analytical method validation. See, the validation experiment should be carried out by an expert analyst to avoid errors due to lack of experience. So see, if you are going to job, obviously your quality can be checked, how expert you are. So for the validation experiments, the analyst should be an expert and he can avoid the errors. Then the analyst should also be very familiar with the technique and operation of the instrument. Obviously, if you are performing a gas chromatographic experiment, obviously there is a need. You have to operate personally this instrument. You have the knowledge of this instrument. So the analyst should be expert and he should be familiar with the technique and the operation of the instrument. Before an instrument starting validating a method, its performance specifications must be verified by using generic chemical standards. And satisfactory results for a method can be only obtained with equipment that is performing well. Obviously, the machine should work very well, then and then only we can get satisfactory results. A special attention must be paid to equipment characteristics which are critical for the method. Means obviously we have to check your machine is working well or not, and you have to take a special attention towards this. For instance, if the detection limit is critical for a specific method, the specification of the instrument for baseline noise and some detectors must be verified. Then see strategies and steps which are involved in the validation procedure. See, first step or a strategy in the validation is develop a protocol, means develop a procedure. An operating procedure or a validation master plan, that is a VMP, for the validation. Means you have the already developed procedure we provides for your experiments. So such a type of protocol can be initially developed by a developed laboratory. This is called as a validation protocol, an operating protocol, or a validation master plan, which is useful for the validation purpose. Then second point is, for a specific validation project, defined owners and responsibilities should be there. So for the validation projects, the owner of this project or owner of this procedure will be there, and he has responsibilities, he has a copyright of that procedure. Then develop a validation project plan. Then fourth one is define the application, purpose and scope of the method. Obviously, the procedure or a method which you have find out, this should be applicable to the human being, to the environment and so on. So initially you have to define the procedure is applicable or not, then purpose of this procedure, then scope of this method. Then fifth point is define the performance parameters and acceptance criteria. Means how you can follow this procedure. This can be initially defined. The sixth point is define validation experiments. We have to define, we have to fix how this experiment can validate. Then verify relevant performance characteristics of the equipment. Then last point is qualify materials, means standards and reagents for purity, accurate amounts and sufficient stability. Means in laboratory you can take 0.3 gram, 0.2 gram. How this can be taken? So this can be initially defined by any scientist. So qualified materials means you have to define how much amount of standards will be added, reagents will be taken, purity of that reagents, accurate amounts and the sufficient stability. All this can be initially defined, and this is a step of a validation. The next step of validation is, ninth step is perform pre-validation experiments. Means before validating this experiment, before finalizing this experiment experimental procedure, we have to already check this procedure is working or not, and then you can say this procedure is working very well. So perform pre-validation of the experiments. 10 point is adjust method parameters and or acceptance criteria if necessary. If in pre-validation some parameters were varying, so adjust the method parameters. Then 11 point is perform full internal and external validation experiments. Then 12 point is develop standard operation procedures for executing the method in the routine. Means you have to develop a standard operation procedure for execution the method. 13th criteria is define criteria for revalidation. Revalidation means, pre-validation means before saying that your procedure is exactly working in your way, before that checking of this method is called as a pre-validation. And after validation, suppose there will be some incorrectness again, then this can be revalidated. So define the criteria for revalidation. Then 14 point is define type and frequency of system suitability test or analytical quality control checks for the routine. Then 15 point is document validation experiments and results in the validation report. So all the parameters you have to check initially, and then you can see your procedure is validated, and thus all the documents can be collected in one report, and this report is called as a validation report. So next point is, based on the experience of the following sequence has proven to be very helpful for the validation of any procedure. So this definitions you have already seen in the chapter number 2, but I am saying all this here because this all 5 parameters are very important in the validation procedure or validation method. So first point is of specificity. Specificity is optimizing separation and detection of standard mixtures if selectivity is insufficient. Means optimize the separation and detection of standard mixtures. Then second point is linearity, range, limit of detection and limit of quantification. All these terms are very useful. You know the definitions of all these terms. Then third point is a precision, means repeatability, reproducibility. Then fourth point is accuracy at different concentrations. Fifth one is a robustness. Robustness means remains unaffected by any change. So this all the parameters are very helpful in the validation procedure. Then some of the parameters can be measured by combining experiments. For example, when the precision of peak areas is measured over the full concentration range, the data can be used to validate the linearity. Means simply precision of peak area is measured over the full concentration range. Means you have prepared 0.1, 0.2, 0.3, 0.4, such a type of series of a concentrated solutions and your readings were repeatable readings, reproducible readings, and this data can be used to validate the linearity. Means y is directly proportional to x or not. This can be checked, and so the validation can be done. Then during method validation, the parameters, acceptance limits and the frequency of ongoing system suitability tests or quality control checks should be defined initially, and criteria must be defined to indicate when the method and the system are beyond statistical control. Means during a method validation, the parameters, as usually I have said, the specificity, range, limit of detection, quantification, accuracy, precision, and so on. So these parameters are important. Then acceptance limit and frequency of ongoing system suitability test. And this can validate a method. Then documentation, which is useful for a validation. So validation protocol is the one. Depending upon the culture of the company, a method validation protocol could be simple or exhaustive. How solutions are going to be made, the experimental design, how the calculations are going to be performed, any software to be utilized as Excel. So see how the validation protocol can be prepared. So depending on a company culture, a method validation protocol will be simple and exhaustive, because how the solutions are prepared, the experimental design should be designed very well, and how the calculations are going to be performed, or any software can be utilized for this performing a plot. So this all the parameters are initially important to make a validation protocol. Then if a full length protocol is required within a particular company, then the writing of this protocol and approval of the protocol would need to be completed prior to the commencement of the validation work. Means simply, if any company requires a full length protocol, means a complete protocol, then the writing of this protocol and the approval of this protocol would be needed to be completed prior to the commencement of the validation work. Means protocol writing and the approval of that protocol is a necessary thing for a particular company. Then otherwise, if this protocol is not validated or the not a approved protocol, then there may be many deviations to the protocol which will be needed to be referenced in the final method validation report. Some companies also have templates for the validation reports, thereby allowing the facile population of the results. Once populated, the file is reviewed to determine if all validation parameters and acceptance criteria were met. If they were not met, a deviation is added and the proper justification must be given. So see, initially we have developed one procedure. This procedure is validated by someone initially. Means validated means what? That checked all the parameters which written in the procedure can be okay. If okay, then this procedure can be sent for a approval. Suppose this procedure is approved, then you have to use this procedure as a validation protocol. If the protocol is not validated, then or some corrections are there, so you have to check the parameters which are not correct or not met with the written parameters, then some new parameters will be added or changed, and some deviations can be there as a positive deviation or negative deviation. Means you have to increase some amount of reagent, decrease some amount of reagent, change temperature either higher or less, change pressure. So each and every experimental parameter can be changed, and then you can say your procedure is working very well. You can give a proper justification. Then, if the analytical method has not been approved at the time of writing the validation protocol, it is recommended to attach a final draft of the method to the protocol. Means, suppose the procedure which is written by you is not approved at the timing of writing the validation protocol 8, then they recommends you to attach a final draft of the method to the protocol. Before starting the experimental work, the protocols must be written by a qualified person and approved by a quality assurance department. Means you cannot directly use the procedure which you have developed. So this procedure can go through many steps, and then only this procedure can be validated, and then this procedure can send to the approval, and when this procedure is approved and this protocol can be approved or developed only by a qualified person. Some of the items that are necessary to be specified in the validation protocol are as below. So, in the validation protocol, some necessary points can be written as here. The analytical method for a given product or drug substance, then the test to be validated. Next one is the test parameters for each test, including type and number of solutions and number of tests. The acceptance criteria for each parameter based on an internal standard operational procedure. Then list of batches of drug substance and or drug products. Then for drug product, the grade or quality of the excipients used in the formulation. Then list of reference materials to be used in the validation experiments. Then information of the instruments and apparatus to be used. Then responsibilities as author, chemist, analytical research project leader, and the quality assurance. So see some necessary points which are included in the validation protocol as obviously the method which is developed is useful for a product or drug substance that can be determined initially. Then the test to be validated. Then the test parameters for each test, including type, number of solution, number of tests that can be written in the validation protocol. Then the acceptance criteria can be written. All the parameters required for the internal standard operational procedure can be written properly in the validation protocol. Then how many batches of the drug substance or drug products that can be listed. Then the grade or quality of the drug product can be written. Then the reference material which is required for the drug preparation that can be written in the validation procedure or protocol. Then the information of the instruments and the apparatus can be written. Then responsibilities like author, chemist, analytical research project leader, and the quality assurance department that can be written in the validation protocol specifically. Then last point is a validation report. So validation report is the main point which is used as a reference, and it has a specific code number used as an identifier within the pharmaceutical organizations and the corresponding drug substance or product name. Note that for early phase method validation reports, the results may be filled in a predefined table and compared with the acceptance criteria. So, in the early time, the phase method validation reports can be prepared and the all data or the result can be filled in a predetermined table or a chart and compared with the acceptance criteria. Means your results can be compared with the reference. Then, however, for late phase validation, means after phase validation, more explicit reports are generated by explaining every experiment with detailed steps of a sample and standard preparation. The list of reference materials as well as the list of the standard or any compound which is required can be reported there. Then the calibrated and the qualified instruments can be used for a validation experiments, and that should be written in the validation report. Then for a drug substances, the list of the batches of the drug substance, notebook number, reference number for any individual impurities or solutions or used intermediate should be noted or listed in the validation report. Means each and every small parameter can be noted in the validation report. Means drug substance, how many batches of this drug substance, notebook number, reference number, impurities, solutions or intermediate that can be listed specifically in the validation report. Then for drug product, the list of batches of drug substances, drug product and the grade or quality of the excipient should be listed there. Then the test parameters and acceptance criteria must be listed together with the results for each test, and the passed or failed results should be indicated. I mean, this procedure is working or not. This can be noted in the validation report. Then the validation report should also contain whether the method validation was successful and if any changes should be there in the analytical procedure or in the analytical method, then the final analytical method must be resubmitted for quality assurance approval. Then once the method is validated, this can be transferred from one laboratory to another laboratory. So the method, when validated, then the validation report should be prepared with the following informations. As already I have told, objective and scope of the method means applicability, type of the method, summary of the method, then all chemical reagents, reference standards, quality control samples with purity, grade, their sources, detailed instructions on their preparation can be written in a validation report. Their procedures for quality checks of standards and chemicals used on that can be written. Their safety precautions, method parameters, critical parameters from robustness testing, listing of equipment and its functional and performance requirements, cell dimensional, baseline noise and column temperature range. Each and every parameter can be noted in the validation report. For complex equipment, a picture or schematic diagram may be useful, means how to handle this instrument and so on. Its guide will be there. Then detailed conditions on how the experiments were conducted, means procedure can be written in very detailed format, including sample preparation. The report must be detailed enough to ensure that it can be reproduced by a competent technician with compatible equipment. These statistical procedures and representative calculations and procedures for quality control in routine analysis. Example, system suitability test. Means each and every parameter can be written. Procedure is there, how to perform this experiment, how to calculate each and every parameter, how to prepare solutions. Each and everything can be written in a report. Then plots should be given. Then method acceptance limit, performance data, the expected uncertainty of measurement results, criteria for revalidation, references will be given. Then summary and conclusions will be given. Then approval with names, titles, dates and signature of those responsible for the review and approval of the analytical test procedure can be written. Means who has approved this procedure, this can be written, the name of that scientist, title,